A plasmid is a small circular DNA molecule found in bacteria. Plasmids are a kind of portable set of genes that bacteria are able to exchange with each other. They can mutate more rapidly than the main bacterial genome, and as a result can confer new traits to their host bacterium. If a new trait appears, for example resistance to a drug, then this trait will be passed on to all offspring and can be exchanged with other bacteria, even of a different species. The discovery of plasmids led to genetic engineering. Plasmids are active gene transcribers in bacterial cells, so if a new gene is added to the plasmid, a donor gene, the product of that gene can be produced by the host bacterium. In this method, a plasmid is isolated from bacteria. It is then cut using a restriction enzyme. A new gene from any living source can then be inserted into the plasmid using DNA ligase, another enzyme. This newly stitched up plasmid is then inserted back into the bacterial cells, a process called transformation. Plasmid-containing cells are then artificially selected and cultured. These cultures produce the human gene product, which can then be purified. Of course, nowadays, the plasmid is not obtained anew from bacterial cells. There are many different plasmids available from biotechnology suppliers. They're all engineered to have useful traits, such as the LAC-Z gene or LAC-Z gene, which turns the bacterium blue if cultured in a particular medium. The inserted gene interrupts the LAC-Z gene, and thus white colonies have the gene successfully inserted. Commercial plasmids are of course completely sequenced and mapped, so the genetic engineer is able to select from a wide range of restriction sites. This is important because the donor DNA may not have many compatible restriction sites. So it may be just a small circular piece of DNA, but it is the cause of, and the solution to, many of our medical problems.